this is an example lecture which I took from my non-major biology classes. So you see it's a 100er, so basically a first year course, or for everybody who's starting with science and want to have a peek into biology to explore if this is something for them. So those can take this kind of lecture. And yes, the topic which I will talk about is a little bit about microbes. So one of my favorite topics, because yes, I'm also doing research on this. And yes, you see here, this is basically a picture from parts of our gut. You see a lot of food particles there, and then a lot of tiny organisms, so microbes basically, which help us digest our food. But we are coming back to this later. So, Nicole Hansmeier. I'm an assistant professor here at the college, and my lovely accent as well as the name, yes, it comes from the old word. Precisely, it comes from Germany. So if you want to learn some German, please feel free to stop by. I'm happy to chat with you in German also. So what can you learn from me, or what kind of courses do I provide? I give introductory courses for biology, for the majors, but also for the non-majors, which you can take with me. Then I focus on my main courses are all around medical microbiology, an introduction or an advanced one. So where we talk about basically pathogens, our immune system, the interplay, how we can battle our pathogens or our beneficiaries, which we have and grow inside of us. We will talk about what kind of repertoire of pathogens we have there, how they trick, and how diseases are developing. And you can see, for instance, one example is here on the right side. This is a pathogen underneath in the red-orange color there. This is a eukaryotic cell. And this green pathogen, this is a Salmonella, which is docking on side of our cells there and trying to make their entry inside there. And if you are really interested about this, so please feel free to ask questions or join some of my lectures for this, because yes, we will go into detail how the pathogens are basically getting inside, battling, and hopefully not succeeding with developing a disease. Other courses are about research then, so undergrads and also do research with us. So join me in the lab, and also my students. There you can basically see how pathogens are invading our cells and do these kind of nice images, what you can see here on the right side, or also videos, how they do this. So courses for this are, for instance, 396. This is a kind of lab course, and the 498 and 9, those are the bachelor honors courses. If you then still do not have enough from me, then you can also stay with me and also do some graduate studies with me, and there are also courses for those around. So what am I doing when I'm not doing or teaching you about my favorite topics and also the introduction? Then you can find me doing research in the lab. And what am I doing there? So I'm focusing on understanding of human diseases and the effects of exposures. I'm choosing to analyze those diseases system biology approach. So this is more a holistic view on deciphering the complexity in the emergent property of a biological system. So I'm looking on us like on a network, and what happens when I'm pulling on different parts of this kind of network, how I'm interfering and how I'm tweaking and also improving the system by doing so. So I'm manipulating systems, pathogens and host system, and seeing what the impact of this is. One particular focus is what I'm specialized on is salmonellosis. You might not know salmonellosis per se. You might better know it as food poisoning. And yes, you also know this quite often. We have food recalls. So these are food recalls. This one is a recent food recall, basically. Food recalls are often because of basically that food is contaminated, which can be with metal parts, but also quite commonly with pathogens. And Salmonella is one of the pathogens which causes a lot of food recalls. So last year we had, or also this year we had around 10 food recalls already. What kind of products are contaminated? So when we talk about salmonella contamination, we need to think about bean sprout, radish, chorizos, eclairs, chicken, chocolate mix, spices. So a lot of different kind of food can be contaminated by salmonella. And yes, last year we had a really big recall, so big recall of food, of onions, where basically this caused 900 cases in the US and ended up with a lot of hospitalization of persons who consumed this kind of food. And also in Canada, you see here we have 240 illnesses there and also a lot of hospitalization due to this. This year, big one, so you see here, it's from July 30th, where basically Frankie's red hot seasoning is contaminated. So what happens when you eat poisonous foods or contaminated food? Obviously, or quite commonly, this is not lethal for you. So everything is good, breathe. So the only thing is what you can get when you eat these poisonous foods, that you get nausea, get fever, cramps, and followed by watery diarrhea. So you will spend a lot of time in the bathroom. And this basically starts after 6 to 72 hours after your exposure with this contaminated food. How long does it last? It lasts around 2 to 3 days. It can proceed up to a week. Then most of us will recover by ourselves, so without any kind of treatment. The only ones where this disease is more serious is for newborns, elderly, or immunocompromised. Those need to go to a hospital, see a doctor, get antibiotic, and sometimes even intravenous therapy for this. How many cases do we have in Canada per year of salmonellosis? Around 80,000, and around 1,000 persons per year get hospitalized, but the numbers are rising because of, yes, antibiotic crisis. So this is a big topic for the future, where we need to find new treatment options. Another kind of research what I'm doing is cannabis testing. Yes, you all know cannabis is legalized, and also the university gets a research license in fall, so then we can start with looking on different kind of cannabis product to ensure food safety on one side, and also to explore what kind of long-term effects do we have when we consume cannabis. So this is what I'm studying in another kind of project. Last but not least, I'm also involved, and you might have seen this posting on Facebook from Regina. You see updates, weekly updates about our SARS-CoV-2 or COVID-19 or pandemic, basically how much viral load we have in our wastewater system. And so what I'm doing, I'm not following. So this is basically coming from our group. So we are posting the numbers, how the case load or how the viral load in the wastewater is in our city. So we followed it since the last year. So we are basically extracting fecal matter and then analyzing how much virus we have inside there, which gives us, since we are profiling the whole city, an overview how many cases or how much load of virus do we have in our sewage system. So an unbiased survival system, what we have there in place, and we report those numbers to our health authorities, which then combine those numbers with the clinical testing, and this gives them an idea if they are good with the testing or if we then need to increase the testing. So if we see that there is a spike coming up, that they are prepared to what is to come in our city. If you're interested in those, or if you're in general interested to see how lab work is done, where can you find me? So you can find me basically the fall in the 5th floor in the RIC building. When I'm not teaching, I'm up there in my lab. So shared with my colleague here, Dr. Chow. And here are 2 of my students. This is before the pandemic hits us, because yes, now we are all wearing face masks here, but we have lots of fun there also. So if you're interested in this, please feel free to shoot me an email or stop by in the lab that I can show you around. So this is about me. In general, when we start the lecture, I will also ask questions and about you. So who you are, what kind of visions you have, what you want to learn. And from this, I will then combine this information and also integrate this in the lectures in the future, that everybody gets a little bit peek of their interests inside there. So in general, when we have a lecture then coming up, then you will see something like an overview, what this lecture is about. So you will see lecture objectives and what we basically will talk about. And at the end, we will always wrap up this lecture with a lecture review where you get basically questions from me, which I like you to answer by yourself. And these kind of questions sometimes are also popping up then in exams. So it's basically a good study guide for you to prepare yourself for exams. So and here you see basically we're talking about microbes today. Then we're talking about a little bit about bacterial structures and how they share and exchange material, and then what kind of role bacteria or microbes have in our life. I will also point out always in between where you can find additional information for what I'm talking about, so that you can still after the lecture read up on these different kind of topics. So in my lectures, I'm using open source books for this. You will also see that I will use certain kind of examples from other kind of books. Those books, I'm not expecting you that you buy this. You can borrow those books if you're interested in those examples and reading up more about certain kind of topics from the library. So they are all in Luther Library. And all the information which I'm talking about, also all the slides are posted after the lecture on your courses. So following your courses, you can always see what kind of lectures we're on. You can see at the end the study guide. And yes, we also have at the next lectures always a kind of review session where we talked about some of the questions which I posted. And yes, you're always free to ask questions and unmute yourself. So don't be shy. So from our perspective, when we think about microbes, we also always think about diseases. Yes, we know COVID-19, cholera, leprosy, and also this nasty image of flesh opening burn, what these patients had. So necrotizing fasciitis, this is how the disease called. Those stuff we know. Pathogens here are listed below there. So all these pathogens can produce toxins, they trick our immune system, they feed on us, they basically grow on us and establish themselves. But is this all about microbes? Are they all mean, or are there also some good ones? And yes, this is what we will talk about. Before we come to this, let's chat a little bit what microbes are, because yes, it's an introductory lecture, so you get all the basics of also for this. So what are microbes? Microbes are tiny life forms. The word is already descriptive by themselves, microbe. It comes and has a Greek origin, which means small life. And yes, we have them everywhere. And I hope that we're getting back to this kind of scenario. Then they're surrounding us. But yes, we have this community feeling then again. So how small are they? So let's check out this chart here, where we are there. So this is a logarithmic scaling from nanometer to meter. Yes, here's a normal adult there, and here is an atom. So where are microbes located? They're in the micrometer area. So what does this mean basically? Where are we? What can we see basically? So can anybody of you either unmute yourself or point out in the chat, what can we see? So which kind of scale? So where could we identify those kind of objects which are pointed out in this diagram? So obviously this we can see. So meter we can see quite clear. How far can I go down here? We can see up to 100 micrometers. So there is our limit. So everything below we cannot really see. So microbes we cannot see. And to give you another kind of idea how small micrometers are, give you an example here. So this person is 1.7 meter And yes, one microbe, for instance, E. coli, commensal of our gut system, it's 1 micrometer, which is, a micrometer is a millionth of a meter. So you see here to the power of -6 here. So this is still a little bit vague. So not really that we can grab it. So let's put it in another kind of way around. So if the E. coli would be the size of us, so 1.7 meter, then basically we, or this adult, would be basically the size of Saskatchewan. So I think this is now easy understandable how big the difference is there. So, and this big size differences, biology is always working there to show them with exponents, and you will come across a lot of log scales for this. So another question is, when we're talking about microbes, are all microbes the same? Are all microbes basically related to each other? And thus we can say basically, no, they're not. So you all know from school, so the tree of life with different kind of domains there. So we have eukaryotes, plants, animals, and us, we are falling into this domain. Then we have archaea there, and we also have bacteria there. And microbes, they belong to all of these kind of domains. So you can find them in all. And even here, so this is a tree proposed from Carl Wöse. The newer trees, when they are generated basically on genetic information, we can also include, or some of us includes now also viruses inside there. So where do we put this? Close to the origin of life, basically, which we will focus on in another kind of lecture. So they are also belonging to the categories of microbes. So microbes are not related to each other all. So we have them in all different kind of domains. And now we will focus a little bit more on prokaryotes. So basically the domains bacteria and archaea. So prokaryotes, since we are still debating if viruses belongs to our tree of life or not, because some scientists are really against this. And yes, in another lecture, we will also talk about why they're against this. Everybody agrees, at least from the scientific community, that yes, prokaryotes were the first life forms on Earth. So there's not a discussion going on. Another discussion is then going on, okay, when did they appear on Earth? So there we have a range then given here, 3.5 to 4.3 billion years ago. Why do we have this kind of confusion with this kind of billion years of a difference? I will talk next to this because we got some new evidence which makes us believe that yes, the first life form could have appeared earlier on Earth. What happened when prokaryotes came up on Earth and established on Earth? So when was it basically? Roughly 2 billion years before eukaryotes were there. And what happens then when they settled then? Basically, they made it possible that oxygen were accumulating on Earth, and then the origin of the eukaryotes could happen because the Earth was then oxygenated. Before prokaryotes and their activity, no oxygen there, so no good breathing for us there. So they basically transformed the Earth from an anoxic origin to what we have now today. And what is the support for this? Stromatolites. Here we are on the coast of Western Australia, Shark Bay. You can see these kind of mounds here. And when you cut through this, you see these lights there, and those are all fossilized microorganisms. And basically they dated these stones, how old they are, and they came back with 3.5 billion years ago. So where's the confusion now? Because yes, new information came up from colleagues from Quebec here. So a study which is published in Nature, and if you are interested in this, so you see sometimes at the bottom of my slides, there are links to papers or also links to videos here. So here I have both of these. And then in my lecture, I often show also short videos about those kind of new findings or also summaries about our stuff there. And here basically this research group discovered new fossilized microorganism in a hydrothermal vent. And they basically dated it to 4.28 billion years, these kind of structures of this. Here is an interview basically with the researchers who developed or who identified these kind of structures and dated these structures. And they tell you a little bit how fascinated their research were and what they did about it. To go a little bit further, so when we talk about prokaryotes, we are multicellular, so we have a lot of cells which are working together, a lot of tissues, a lot of organs made of a lot of cells. Prokaryotes, on the other hand, most of them, they're unicellular. So that's one cell, and one cell, basically, they can do everything so for themselves. And yes, they're also not all boring and not look all alike. So you see different kinds of shapes here. Those are the three most common ones. So cocci, bacilli, or spirally. But yes, we also have complex mixtures. Basically, we have merging forms between those, which we discovered lately. How we see prokaryotes or microbes under the microscope. But microbiologists, when we study them, we often identify them by their morphology. So basically how they are looking like when we grow them on these kind of plates. Those are agar plates here. And each of these dots, basically, it's a colony built from millions or billions of prokaryotes. So each color has a different kind of prokaryote here growing on this. And what you can see here, this is basically a handprint of a 5-year-old who was playing on a playground. And after this, the researcher asked the child to put his hand on the agar plate. And from this, basically, you see what all was growing on the hand of this young child. So kind of nasty looking here, what you see there, what you can get when you play on a playground. So if you have kids, please advise them after playing there, wash their hand, because then you can get rid of those nastinesses. And the next thing what we will do, and to see if you're still with me, we will have some kind of quizzes here. And here, basically, you see these agar plates from different kind of sources where colleagues streaked out material where they sampled it from. So we have a drinking fountain, we have the lobby floor, we have a kitchen sponge and dirty socks. And what I like to do here now is basically do a Mentimeter where you can vote which kind of plate comes from which kind of sampled source. A was actually the drinking fountain. So kind of good. Only 2 contaminations are on there. Whereas this one, the C and D, as well as the B shows a high complexity on it. Whereas C and D, it's kind of all the same. So when we do this a little bit more smaller again, you see here, it's a little bit looking more alike, more homogeneously, which came from actually one contamination from a sponge and from a dirty socks. Whereas, yes, here's a lobby floor, so a lot of things is going on there. And yes, I would not like to sit on this floor and eat on this floor because, yes, you get a lot of other nastiness with this. Okay, so now we're coming back to a little bit more information. So about prokaryotes. So here you see basically the prokaryotic cell, how all the prokaryotes are looking like, compared to eukaryotic cell. This is how our cells are looking like, or at least some of our cells are looking like. What you immediately can see here, oh, that's really big. And you see a lot of different kind of dark spots inside there, which are organelles inside there. So it looks more complex. It looks bigger and more complex than the prokaryotic one. And also you see this big gray area in the middle there. So this is basically the nucleus where all our genetic material is stored inside there. Whereas when you look over to the small prokaryote, yes, you also have some gray area there inside there, but they do not have a nucleus. They also have DNA inside there. So this is what the gray stuff is about, because they used a certain kind of staining to showcase it. But those kind of DNAs, they are not wrapped up in a nucleus or in an organelle. So they are floating around in this area, in an area which we call nucleoid. So they're completely different looking like. And yes, all these different kind of compartments, they do different kind of jobs so that we can run several stuff in parallel. Whereas prokaryotes, they need to run all the processes after each other. So this is what we have. Our cells do parallel processing, prokaryotes, single processing. So where does the name come from? Here you see the nucleus in eukaryotes. And yes, from the name eukaryotes, it's again Greek again, because yes, a lot of these kind of names came either from Greek or from Latin. Eu means true, karyon means kernel or nucleus. So eukaryotic cells have a true nucleus or are true nuclear cells. Whereas prokaryotes, pro means before. So before there was a nucleus. And then we would dig a little bit deeper inside there, how prokaryotes are looking like, what kind of simple cell structures we can see there. We would go through the different kind of parts here, what they do there, what you can find there, and basically how they're similar or how they're different to eukaryotic cells. We will also point out then a lot of specific structures which we see there, which make prokaryotes kind of fancy to look at, even under the microscope, because what you can see there quite commonly, so some of them have a capsule, it's a sugar coat surrounding them. So why are they good or not so good? They are not so good when we think about our immune system because they protect themselves. So they protect the pathogens from discovery by our immune system. And in the environment, they help them to last longer because they protect them from dehydration. Also fancy stuff like survival forms. So they have these kind of endospores or spores, what they form there. So also basically when everything is gone from Earth, those kind of spores, they're still there. And when they are good condition coming up again, they can thrive again. So this is what they so survival forms for where basically they're inactive at this time point and then thrive again when condition becomes better. And yes, we have those kind of problematic prokaryotes when we think of hospital settings, because there we also have prokaryotes Clostridium, which builds spores, which lasts them there, even when we clean up everything really tight, even with ethanol and all detergents we are going through this, still they will be there, some of them, and then they can thrive again when they found new kind of growth material, for instance, an infected person to grow on. So, and here you see a lot of other stuff, but they have their stuff for mobility, so that they're not only immobile, sitting on one place, that they can colonize all the other stuff, parts for attachment to our cells, and yes, in our gut to settle on them. Then protein coats, which makes them possible to even grow on hydrothermal vents, so or in really hot areas. So, or they have also these kind of stuff where they connect to each other, so which we call a sex pilus. This is not that they are pushing their genetic material through this, it's more like to basically move the other cell close to each other together. So they're quite successful and they're quite abundant on Earth. I hope I could convince you by this. And they also are genetically quite diverse. So they reproduce quite fast, which is also an advantage for them. And when we talk about reproduction, about prokaryotes, this is the keyword you need to memorize. Binary fission, this is how they do this, how we call this process. So what is this process? Basically, this process is to make a copy of a prokaryote. So an identical clone is formed by this. So basically, what do? Basically, we have a bacterium here. They grow bigger. They replicate their DNA inside there, all the different kind of parts of DNA, what they have there. So they have one chromosomes, and here you see they have two kind of plasmids, small DNA fragments where they encode additional information on it. So they're growing big, growing even further. They have replicated their DNA, and then basically they're splitting up in the middle, and then they have a daughter cell. And yes, it can be as fast as 20 minutes. So after 20 minutes, an E. coli cell under optimal condition without any kind of limitations, they can have a double of them. And yes, here is a short kind of video how students in the lab can visualize and see how those bacteria are replicating. So this is why they are so fast, why we see them all around there, and yes, why they are so diverse. This is what we see here. So because they can share genetic material with each other. So here you see a small DNA fragment is given, provided by a donor to a recipient, and thereby this new bacterium also gets this new information. This process we call, you need to remember here, conjugation. Then we have transduction. So also we have viruses which are infecting bacteria or prokaryotes. And those kind we call phages, and they can provide additional genetic material also when they infect the bacterium. So this is called transduction. Another thing to remember. And then we have here, they can also take up DNA from the environment, which we call transformation, and also integrate this new DNA into their genome inside there. So this is how they more diversify even more and how they can evolve quite fast. And this kind of processes allow them to thrive everywhere. So they can thrive in cool temperature. So those we called spherophylls, for instance, here. This is an example plotted here, Spherobacter. They can thrive in hot places, hot vents here. So this is an archeon where they can settle on it. In salty, high saline area. Also, this is San Francisco Bay. But yes, they can also here, this is what we also experience quite a lot here in Saskatchewan when we have our blooms there. So they can grow also on water there, and then they can produce certain kind of toxin, which makes them not so nice for us. But yes, these kind of fast spreading without using any kind of material besides carbon dioxide and light energy, they can grow as big as what you see here, these kind of blooms on top of it. We also have different kind of what they can use and what they can metabolize there. You do not need to remember those. So we have what we call chemoautotroph, where they can use inorganic carbon for this to produce food, heterotroph or chemoheterotroph. And here are some examples for those. Now we're coming back to the beginning of our lecture, because we saw now a little bit about microbes, what they can do and how diverse they are. And now what we will do basically, and we will do this again with a Mentimeter, in class we will do this in a kind of board game or group discussion, where basically I like you to point out what kind of benefits could bacteria have for us. So we already saw the nastiness at the beginning, all the diseases, but is there also something what is beneficial for them? Okay, so this is where I was pointing out your fridge. Think of cheese, beer, sauerkraut, or all the other fermented food or fermented cabbages. There where microbes are playing and are good for us. Yes, you all mentioned our gut system, yes, and antibiotics. You also mentioned there a lot of other stuff we use to produce antibiotics or treatments for us. So we use these kind of organelles for production. And yes, you need also to think about our wastewater treatment plants. So those prokaryotes, which are helping us basically to digest all the nastiness which we release inside there. We're using them also for biofuel, bioenergy, and also biogas. So a lot of things where they can help us. So degradation of organic or inorganic material and clean up all our environment. So they are really good for us as well. So we do not have only nastiness, but also a lot of benefits on the other side. And yes, since you mentioned all our complex microbiota and our gut system, there is no exception. So the microbes play a big role on us, in our bodies, for us. You see here microbes, we have 39 trillion versus eukaryotic cells, only 30 trillion. So basically half of it, of our body is prokaryotic and eukaryotic. So those are estimates from 2015. And the paper you can see here on the bottom where I got the information from. And a lot of when we think of our body, here's basically a map where they took a swab from each part of different humans and then sequenced, so identified what kind of prokaryotes are on there. And from this, they came up with these kind of diagrams representing how many bacteria or which kind of bacteria or archaea are on different kind of sites. And you see that there's a lot of variation going on, but the biggest variation is basically in our gut system, our intestine. What we also learned, since yes, microbiota, this kind of research is kind of hip nowadays, we learned that we change our microbiota over the time throughout our life. So from an unborn one, breastfed one, have different gut microbiota to an elderly. And here, this is where a new study basically is focusing on why certain people become so old. So can get over 100 years old, how their gut microbiota is looking like. So they got some kind of new information about this, about aging and the relationship to the gut, so that we need to treat our gut properly to basically get to this kind of age. We also learned about that we can shape this. So even if our gut microbiota is totally messed up, we can therapy this microbiota and shift it back. So they did this here. Basically, this was a study where they tried to remove the disrupted gut microbiota and transplanted a better looking microbiota where they characterized this before. And this was an example. They didn't do it on humans. This was what their future vision is looking like. That basically we do a kind of gut transplantation of our microbiota and then basically getting a new kind of gut and also a more healthy gut. This is also what the study about aging and looking for 100 years plus they did on mice so far. And they could find out when they transplanted an elderly mice with a new or young mice microbiota, they could rejuvenate this elderly mice. So there are some kind of nice things coming out, which was recently published in Nature. So, and this is basically summarizing also where we found a lot of linkages between aging, metabolic diseases, such as diabetes, obesity, Alzheimer, and atherosclerosis, but also how our lifestyle can impact our microbiota and what we can do to alter it again. So, and this would be then the end of the lecture. So here we would wrap up. So have questions which I like to ask that the students are answering this in their free time. And then we are getting back to the next lecture together where we would then follow up on some of these questions, discuss these questions. And these are basically kind of also help us for when we do the exam, because some of these questions are popping up later on in exams.